Okay, hi guys. Let's go ahead and get started. Hopefully you all can hear me. I'm Kate Williams. I'm one of Dr. Springer's graduate students, and I'm going to be teaching your lecture on wildlife disease today. So for those of you who haven't seen my face or half of my face, I'm a second-year master's student here in the department. With my advisors are Dr. Springer and Dr. Cox. And I focus mainly on elk calf survival here in Kentucky. So we're going out in the summertime, we're catching these little critters and putting transmitters on them to track them through their first year of life. But while we're doing that, we're also collecting blood and hair samples to get at some of the physiology and health of these animals, which is kind of in line with what we're talking about today. And I was going to go into kind of a bit of my background before I got to grad school, but for sake of time, we're going to skip that today with the caveat that if you have career questions, please feel free to come up and ask after or at another time. So today we've got a lot of stuff to get through, so bear with me. We're going to talk about some disease terms and concepts, disease in wild populations, some case studies, uncertainties in surveillance and management, and then finish up with some implications for humans. And also, I'm not used to talking in front of a big group, so if you can't hear me at any time, please just let me know, okay? So let's start off with some disease terminology. The most basic one, what actually is disease, right? This is a change in the normal structure or function of an animal and generally excludes physical injury. So if a deer gets hit by a car, that's not generally disease, that's just trauma. And there are two main phases. So the first is subclinical. This is from the time of exposure to the onset of visible symptoms. And it has a couple of different names that you may have heard of before. Then that second phase is going to be clinical disease, or the appearance, which starts at the appearance of visible symptoms. And these symptoms can range from mild to severe or fatal. And one of the big components here, disease always requires exposure to a causative agent. That's a big one here. But that doesn't necessarily mean that that is the only requirement for disease to occur. And that looks like the epidemiological triangle or triad, kind of a mouthful there, but it has three main components, hence the triangle, three sides, right? The first is a susceptible host, and this can be influenced by life history, genetics, stress, which can impact immunity, age. That's a big one, right? So younger animals may be more resilient than older animals. And it can also be influenced by things like population, density, habitat, and nutrition. The next part of this triangle or triad is the environment. So this is things like climate, abiotic conditions, so precipitation, sunlight, things like that. Also, a big component is physical barriers and habitat fragmentation. So those things can help promote or can also inhibit animal and pathogen movement. So those are big ones that kind of set the stage for disease. And then the third one you've already heard me mention is a causative agent. So that's the actual disease causing pathogen or what causes disease. So that can be influenced by pathogenicity, which is how well a pathogen can harm its host. It can be influenced by prevalence or pathogen load, as well as that particular agent's life history and source. And each of these by themselves is not enough to cause disease. So you have to have all three before it can actually occur. Once we have all three, We follow a general disease timeline. This would be true for animals or humans, right? So we start with this initial exposure of a previously healthy animal, which moves us into that subclinical disease phase. We see symptoms appear, which is the body fighting off that pathogen. That's the reaction. We go through that stage of symptomatic disease and then can either go to recovery or to death, depending on how successful the body is. And animals can move through this timeline in a couple or three different ways. So we have acute, that is the severe and sudden onset. So an animal will move very quickly through this timeline. Too soon. We have chronic, which is when a condition is cyclical or lasts for longer than 1 year. So they will move very slowly or kind of get stuck between some of the phases. And then lastly, we have degenerative, which is a progressive and irreversible decline. So they will kind of deliberately march through this, this timeline, and it will always end in death. Let's move on to some disease categories. There are 6 main categories here that hopefully you guys have all seen before or at least heard of. The main 2 are infectious and parasitic. So these would be things caused by bacteria, viruses, parasites, and this is what's commonly referred to as pathogenic disease. But we have some other categories too, including toxic. So that would be due to pollution or chemicals. So think things like lead poisoning. Nutritional would be exactly what it sounds, a deficiency or imbalance. That could be too much iron or not enough iron, things like that. Congenital, an animal is born with a genetic mutation or malformation. And that's the key here, is that it's born with this. So this is things like skeletal malformations, heart defects, things like that, and generally results in a shortened lifespan for an individual. And then lastly, we have physiological, which is a mutation or is a condition or malformation that interferes with normal bodily function. You might say, well, that sounds like congenital, and it does, but the key is that the animal is not born with this. So this is going to be things like glaucoma, arthritis, any of the old people itises, things you develop. And these can be transmitted by a number of different modes of transmission. We have direct contact, airborne, droplet, vector-borne, so being passed by another living organism, and then common vehicle, which would be things like blood, urine, saliva, common methods. And they generally follow the same kind of transmission cycle. So we have our infectious agent, which replicates and reaches a certain threshold in our infected individual or reservoir. And at a certain point, it surpasses that threshold and moves to an exit portal. So something like the mouth or the nose, where it is then transmitted somehow to an entry portal on a susceptible host, where it will then replicate and the cycle can start all over again. And disease transmission is influenced by a number of things, including contact rate. So animals that are social or live close together will have either a quicker or more often, or this cycle will occur more often because they're closer together, so a higher contact rate. The susceptibility or immunity of individuals, and then also the force of infection or how quickly a disease can be acquired. And then finally, I know I'm kind of running through this, so stick with me, guys. I have a couple of just general epidemiological terms that you will often hear in relation to disease. The first is prevalence rate, which is the proportion of the population with a given symptom or disease. This is generally reported as a percentage. And then the last two here, morbidity and mortality, are ones that might commonly be confused because they kind of sound similar, but they mean very, very different things, right? So morbidity is the state of being symptomatic from a given disease, whereas mortality is death from a disease. So big difference there, right? Okay, I know that was a lot, but hopefully you guys have seen, how many of you guys have seen some of those terms before? A lot of you, okay, good, good. Any questions before we move on? Yeah.
Do you know if this PowerPoint is on the, it is.
Thank you, because I didn't know that. So yes, it is. The PowerPoint's online. Yeah, sorry, lots of information. Okay, good deal. Let's go ahead and move on to talking about disease in wild populations, right? So we're just kind of generally talking about disease. We're going to move on and narrow it down. And before we move on, I want to just kind of define what I mean when I'm talking about a wild population. You guys probably already know this. You're in a wildlife management class. But when I talk about wild individuals or wild populations, I'm referring to two sources of individuals. We have wild individuals, which are animals living outside direct human control. And then we also have feral individuals, which would be domestic animals that have escaped and become wild. So there's two kind of directions this could come from. And I'm hoping you guys know that all wild populations have 4 basic survival requirements. Do you guys know what those are? Cover, yep, that's one. Food and water, yep. So they mean it's 4, yeah, I heard it, space, good job. So yeah, there's 4 basic requirements, and keep those in mind because that's going to come into play in the next couple slides. Whoop, I messed up my timing. You get a preview there. So let's start out by talking about disease in normal wild populations. And what I mean by normal is that all 4 of those survival requirements have been met. They have adequate food, water, cover, and space. And when this happens, disease is kind of considered just a natural phenomenon. It's generally cyclical and self-regulating. So you see our preview here. It generally follows population trends, right? So as individuals increase and decrease, so will the amount or availability of pathogens. Because disease in a normal population is enzoonotic, meaning it occurs regularly in that area, that species, that environment, and generally results in compensatory mortality. So it doesn't go above a certain threshold that would cause a population to crash. It's just kind of a normal source of mortality. But not all populations are normal, right? We often have stressed wild populations where the survival requirements are not met because they either don't have adequate access to those 4 basic survival requirements, or they're severely limited. And this can be for many, many reasons, including things like the global wildlife trade, climate change, global trade and transport, right? So you can be in one country one day and be on the other side of the world the next day. Increases in industrial pollution, interactions with livestock, and then the most important one down here, which is land use changes due to expanding human populations, right? So you probably heard about deforestation to turn into farmland or development. That's a big one because it promotes stressed wild populations where disease is artificially inflated, meaning that cycles, that normal self-regulating cycle may be altered and may require human assistance to bring it back to normal, because disease is now epizootic, key epi. So meaning it's more eruptive and can produce more devastating outbreaks, leading to potentially additive mortality. So we've now reached a threshold of more, or beyond that threshold of normal mortality, and populations will crash. Okay, I'll take a quick drink of water here before we move into our case study. Okay, how are we doing so far? Hopefully nothing new, right? Good, good, okay. Awesome. So our case studies, I've kind of previewed them for you. The first one is botulism, two types, C and E, which are commonly referred to as avian botulism. As you can guess by the avian, it affects a number of species of wild birds, but particularly wild waterfowl and shorebirds, and is produced by a bacteria or a toxin in a bacteria that occurs naturally in the soil and in the environment, which is why it's distributed across all continents, but very much more prevalent in North America, especially in the late summer and early fall, because that is one of the prime conditions, which are low water levels and high water temperatures, promote the bioaccumulation of this toxin. So we start out, it's been accumulating in aquatic kind of microinvertebrates, macroinvertebrates that then die because the water levels are too low. Those dead invertebrates get eaten by ducks, and it bioaccumulates in that duck, which then dies and is consumed by maggots, which then takes up the toxin. And then that gets eaten by ducks. And then you can kind of see the rolling stone cyclical nature here, until eventually it accelerates so much that it causes a mass die-off, which is why we care about it, because we see these mass die-offs as a major risk to endangered species. So species that there aren't very many of them, mass die-off could be really bad, right? And it can also be a significant source of mortality for migratory birds as they're moving from one place or the other. So when it occurs, we have some classic clinical signs, including paralysis, muscle weakness, and incoordination, which generally leads to the inability to fly, followed by respiratory arrest, and then eventually death. And unfortunately for surveillance, most birds are found at the death stage. So it might look like this on a beach. You just find a bunch of dead birds and you go back and you do a necropsy and you don't really find anything significant. There aren't very many lesions, and they're generally in good body condition, right? Because they've been eating. So we really have to diagnose it based on laboratory tests where they isolate that toxin in a tissue sample. Now, once they get to the dead stage, we can't do a whole lot for them, right? But if it's a mild case, meaning they're still in the muscle weakness phase, we can remove them from the contaminated environment, provide supportive care, and administer antitoxins, and hopefully they should get better. However, on the population scale, that's not often possible, right? You can't do that for millions or hundreds of birds. So we revert to population level management. That's a key difference here between treatment and management. Treatment is the individual, management is the population level. So at the population level, we can't do eradication. We can't get rid of it because it's a natural occurring bacteria and toxin. But managers can help stabilize water levels and temperatures to help prevent those die-offs. And then we can remove carcasses once we find them to eliminate or break that cycle. Okay, moving on to our next case study. We have chronic wasting disease, or CWD, which I'm hoping all of you have heard of. I'm seeing lots of heads nod, good, good. This is a disease that affects members of the deer family. It's caused by a transmissible spongiform encephalopathy, or TSE, which is caused by a misfolded naturally occurring protein, or a prion. This is very similar to things like mad cow disease or scrapie, which occurs in sheep, and is found throughout North America as well as internationally in Norway and South Korea in captive cervids. It occurs year-round, and you'll see why in a minute. And we care about it because it is 100% fatal with no known cure. That's very bad, right? We don't want that. We want to be able to figure something out. And you'll notice too, the reason I said that we care about that is because map's probably a little blurry, but here's Kentucky right here, and we have successfully kept CWD out. But you'll notice a lot of bordering states, it's popping up, which means it will eventually be here. Yes, go ahead.
Did it originate in North America?
It did, yep. So kind of the epicenter would be around Colorado, Wyoming is where it first popped up. They're not really totally sure what started that initial switch to cause that misfolding. But yeah, it did show up here and then spread kind of outward.
Where the red and yellow areas?
These are, let's see, the yellow are captive facilities. Hold on, sorry, the red are in captive facilities. Oh, yes, sorry, my bad. The red and yellow dots are all captive facilities. The yellow are ones that have been depopulated, meaning they're no longer existing. They've killed off all the animals to try and prevent the spread. And then red are ones that are existing, so they might still be in the process of management or doing further testing. And this map is very current. So this case literally just popped up in September, I believe, in Tennessee. Okay, so the clinical signs. There's a long subclinical phase. So there's a long asymptomatic phase where animals could be walking around and shedding this protein without us even knowing it. But generally, once they become symptomatic, they very quickly digress through emaciation. So they're all very skinny, erratic behavior or neurologic irregularities. A lot of these can just walk right up to them because they can't really move very well. You may also see salivation or drooping ears, hence chronic wasting disease. So they're wasting away. And unfortunately, surveillance, there's many options, which is fortunate. So we can do opportunistic or targeted sampling. And we can do live tests, but unfortunately, that's where that part comes in. They're unreliable. Really, the only reliable test to confirm the presence of CWD is after they die. And that occurs in lymph or brain tissues that gets sent off to a lab for diagnosis. As I mentioned, there's no treatment. There's none at all. Supportive care will not help this animal. They will not get better. So there's no individual level response, but at the population level, we can do a couple things like regulate the import and export of carcasses and live animals. We can reduce population density. We can prohibit baiting, which would increase contact rates. And we can continue working on our diagnostic research, surveillance, things like that. There is no possible eradication because it also persists in the environment for a number of years through those common vehicles like saliva and urine. So we not only have direct contact passing this from animal to animal, but they can get it from their environment. Our third case study is ophidiomycosis, also known as snake fungal disease. This can occur in a number of wild and captive snakes and is particularly bad in venomous snakes, so rattlesnakes, copperheads, things like that. It's caused by a fungus. You'll never have guessed that. And is found now in at least 38 states. So you can see there's a pretty rapid progression here in not that short a time, or not that long a time span, 2006 to 2019. That's pretty bad. And it's mainly seen, or cases are mainly diagnosed when snakes are active, so when it's warm out. and is a very, very significant. So why we care about it, why we care about it is because it can be devastating to snake health and population persistence. So some populations that are already stressed out from habitat fragmentation and human causes can have pretty high mortality rates. So the clinical signs, we often see accelerated skin shedding cycles. We may see flaking or crusty scales. Here, the pictures might be kind of hard to see, as well as disfiguration of the affected areas. And these kind of swelling things can happen due to trauma and some other diseases too. So when we're looking for this, we really need to swab the affected areas in these suspected cases, which are then sent off to a lab to be cultured. Yeah, go
I
ahead.
think you can turn off the lights and so
Oh.
you can see the slide better.
Perfect. Okay, is that better for everybody? Okay. Sorry, guys.
That's all right.
I'm a newbie. Perfect. Okay, so now you can hopefully see these a little better. So that's what the disfiguration and the kind of crusty scales. They might get kind of a cloudy eye or abscesses on their face. But yeah, so you will swab these for surveillance and send them off to be cultured at a lab to confirm that it is in fact that specific fungus causing the disease. Once we do that and we know that it's snake fungal disease, treatment at the individual level may include administration of an antifungal, as well as supportive therapy if you still have that snake in hand, or say it's captive, so you always have that snake in hand. At the population level, again, you're probably noticing a common theme of no eradication. This is another one that can occur in the environment as well as from direct contact. So it's really hard to get rid of something that persists on the landscape by itself. So we can't eradicate it, but when we have those snakes in hand or we're moving between handling snakes, we can disinfect capture and holding materials. And then of course, we can continue surveillance and research to see how prevalent it actually is. Okay. Continuing our quick march here with our last case study. One I'm hoping you all should have heard of before, rabies, right? Okay. If you haven't, you need to. So this is affects mammals, especially the common vectors of raccoons, coyotes, skunks, bats, and foxes. It's caused by lactoviruses, and there's different strains depending on the vector. So it's all still the same virus, but just some different components. It's distributed on all continents except for Antarctica, because there's not a lot of mammals there, and can be found year-round. We care about it because it's a public health concern, and based on everyone knowing about it, that's why, right? It's all concerning to all of us because it's zoonotic. It can be passed from animals to humans, and for animals, it is 100% fatal after clinical symptoms show up. It's a little different in humans. We can kind of treat you and give you some vaccinations to help with that, but in animals, they're done. Clinical signs, so they occur very, very rapidly once they have been exposed to this virus, and it causes behavioral changes due to swelling of the brain, encephalitis, which can lead to problems with balance, aggression, salivation, agitation, lack of fear, and then eventually can cause a coma due to kind of a muscle paralysis and death, which unfortunately is in line with our surveillance, which the plan right now for surveillance pretty much is just euthanasia of a suspected individual and collection for laboratory testing, because the only way to detect it is in the brain tissue. So an animal obviously has to be dead, cut off its head, look at the brain, and then they kind of isolate the virus within it. Because of this, we don't have any treatment for animals, 100% fatal, remember. But we can do some things at the population level, like reducing contact or risk to people and domestic animals, right? So your dog gets vaccinated, people can get vaccinated, and we can wear gloves and safety glasses and masks and things when handling suspected cases. And on the landscape, there's actually been some success with oral vaccine programs. So the National Rabies Management Program through the USDA has been pretty successful with some of their campaigns. You can see them here in yellow. And they haven't necessarily gotten rid of rabies in these areas, but they have rather been deployed strategically to prevent the expansion of certain strains of this disease. So you remember back here, we have, right here is raccoon rabies, and then over here we have skunk. So that oral vaccine kind of line here is to prevent the expansion of raccoon rabies. And here's what these baits look like. They might put them in, fish scraps or whatever, and then they put them in little tubes, or they may drop them out of planes. And then hopefully they'll be taken up by critters and prevent that expansion. I don't have information, and I'm not going to go into each one of these, but these are some other notable wildlife diseases. And this is just North America. And this is just some of them, just to give you a taste. There's a lot to be concerned about. And many of these are up and coming. I am going to point to coronaviruses, which should sound familiar. Rabbit hemorrhagic disease has become a big thing in the Western US. As well as chytrid fungus is a big one worldwide. So,
How many of these are transmissible humans?
oh, that's a good question. See this on this list. So plague and avian influenza would be, mange can be, tularemia can be, coronaviruses, brucellosis, and West Nile virus. So over half, just on this list. Good question. Any questions, other questions from here? I know I've been kind of really flying through this material, so. Good. Okay. Good deal. Okay. So let's move on to talking about some uncertainties. There's a lot of that concerning wildlife disease for many different reasons. So getting into the first one, it starts with us investigating wildlife disease. And one of the main takeaways you can get from this presentation is that we don't often get a complete picture, like never get the complete picture of wildlife disease. And I kind of got a little silly example of this. We have a squirrel population. You guys have seen those all. And it's not uniform across the landscape, so mimicking a true wild population. And in an ideal world, we would know what happens to every single individual. We would know how they die. We would know when they die. So in an ideal world, we would know each of these squirrels died, and maybe it's the red ones are diseased and the purple ones get hit by a car. And we would know all of that. But it's not a perfect world. So we don't get the whole picture. And that's mainly due to two different types of interference. So we have biological interference and anthropogenic. Biological interference includes things like visibility. So it may be difficult to find all of the diseased animals or to see them repeatedly to track the clinical phase or what happens to them. So we may see an animal early on, but never know what happens to it. Carcass condition. By the time we get to an animal that died from disease, maybe it's been scavenged. So there's no tissue left, it's just a skeleton. Or maybe those tissues have degraded too much that we can't actually do any diagnostic from it. They're too degraded. Anthropogenic interferences might be inconsistencies in reporting. So maybe it's a weekend, so no one's working, so we don't hear about any over the weekend, but really these 3 squirrels died this weekend. It may be expensive, so we can't test, unfortunately, every animal that we get for disease. It costs a fortune, not only in the diagnostics, but in shipping them. So it's too expensive to test every animal. And people that collect the animal may not have the expertise or information to deal with that. So maybe it just looks like they got hit by a car. So we're not going to pick it up because we just assume that that's what it is, not that maybe it had pneumonia that caused that. So bottom line, we have our squirrel population, but maybe we only see this part of it because it's on campus and we can collect those ones. We don't know about the other ones. So we're getting an incomplete picture. That's the first source of uncertainty. The second one comes when we consider diagnosis and treatment. And to illustrate this, I'm going to kind of compare domestic to wildlife. So in domestic animals, when we're considering diagnosing or treating infected individuals, we know that they're owned generally by one individual or corporation or whatever. So we know who owns them and we know where they are because they're in a barn or a fenced in area. So that means it's a closed population, right? They have 10 animals and they always have 10 animals. Livestock are easy to approach, right? You can generally walk up to cows, to sheep, to, there's a few exceptions of really mean ones, but you can walk up, which means you can easily treat them or collect repeated samples for diagnosis. And also the owner, because it's one owner, can help to limit contact with other animals or populations, helping to quarantine or contain that disease. So now turning our focus to wildlife with these in mind, I want you guys to kind of help me brainstorm, keeping these in mind. I gave you some hints. What might be some considerations for diagnosis and treatment in wildlife? And I'd like to write these up on the board if you can. Hopefully this thing will follow me. I have talked enough. I'm sure you've noticed. So. What can you guys think of? What might be some problems for treating or diagnosing wildlife? Yeah, go ahead.
They have a larger range.
Yeah, larger range. So they're not confined. Sorry for my terrible handwriting. What else? Yep.
They're high contact, like they are constantly in contact with other species, like they're not separated like a domestic animal.
Right, good. Yeah, so not separate. Okay. What else? Yeah.
Most times they're skittish.
Yeah, right? How many people can just walk right up to a wild deer? Yeah, believe me, I've tried. It doesn't work.
Unless it's got brain.
Someone's been working in the southeast, yes. I think I spelled that right. Skittish. This is a terrible marker, by the way, so hopefully you guys can see that.
One up there.
Ah,
I don't know if that one did better.
they're hiding it. Let's see if that's better. Oh, they knew. Okay. Okay, I think that all
Oh, yeah, that's
guys.
another one. Access to habitats. That might be tricky to get to.
Okay, yeah. Okay. Any others?
I don't
don't know if this would fall under not confined, but it's an open population.
Yeah, it is, which means that we have no idea how many animals we're looking at in response to disease outbreak, right? And those animals could easily walk off somewhere else. It's not confined, it's not closed, it's not known. Yep, good. Any others you guys? Yeah.
It's not owned by anyone. Or
Well, it is, but it's not owned by one specific person, right? It's owned by everyone in this room. It's owned by everyone in the country or in a state. So yeah, ownership is tricky. Okay.
Speaking of ownership, there's property ownership, you might not have access to some of the territory or habitat because someone else owns it, and they're not gonna let you get on it.
Yep, no, that's good. Yeah, so access, so we'll say,
Property
landowner.
ownership.
Yep, no, that is a big one, right? How we manage that on a larger scale, yeah. Okay, I think we have hit almost all of them. Is there any other ones people can think of? Yep. Yeah, there you go. So yeah, some of these species, we don't see them that often, right? So how do we know how disease affects them? We don't know that much about that animal. So yeah, life history. Okay, good job. Okay, I think we hit most of them. We hit all of them. Right, so really what it boils down to here is that domestic livestock, while difficult, is by far easier than it would be to treat or diagnose in wildlife. Put this in its proper habitat. Yeah. Okay. Good job. These uncertainties in investigating or finding wildlife disease and then figuring out what it is and how to treat it have influenced how we view disease throughout history. There was a time where we didn't know what it was. It just happened. They disappeared. It was magic. But somewhere around the early 1900s, we started to notice it. And we developed kind of this aware but fatalistic perspective that disease happens, it exists, it's natural, but that's it. So we didn't do anything about it. We just kind of said, yeah, it happens. Then we had some technological developments. We started to pay a little bit more attention, and we moved into this perspective of concern or frustration, where we're noticing mass die-offs, we're seeing disease events, and we're concerned about it, but we still don't know what to do, so we don't do anything, which is not good. But again, we've had developments in technology, we've had better surveillance, so we end up moving to this control perspective or firefighter perspective, right? We're trying to put out different events. We start to see the development and implementation of planned responses when we see die-offs. So we're starting to do something. We're having some success with those, and we're trying to gauge what works and what doesn't. So we're throwing darts at a dartboard and seeing what sticks. We are still primarily in this control perspective as wildlife managers. We're still seeing what works because we're still learning a lot about most diseases. There really aren't any that we know everything about. There's maybe very few. So we're just trying to control them at this point. But we're starting to trickle into a new perspective and the kind of goal perspective, which is prevention or problem solving. So we're starting to have more organized and effective reactions to die-offs and to diseases in general. But, and we're starting to kind of get into this disease prevention role, but we're not there yet. It's not mainstream. And we haven't figured out fully how to prevent it on a large scale. Also, we're starting to see disease surveillance becoming more routine in general wildlife management. So that's another way we're moving towards this perspective, but it's not routine everywhere. So like I said, we're not there yet, but we're getting there. So we're, it's hard to figure out wildlife disease. There's a lot of uncertainty. We're still not sure what we can do in all instances. So why the heck should we even bother? Why should we care? Why is it important? My response to this is it's very important because it can increase our knowledge of those diseases. So it's the only way we're going to get better is if we know more about it. It can help inform population demographics. So if we know the sources of mortality, we can more accurately map the total amount of mortality and thus know more about how those population cycles work. We can evaluate environmental conditions because maybe something's causing this. So we need to look at what's happening here. Continued surveillance and research can help us improve our diagnostic tools so we can more quickly and more easily identify what's wrong. And we can work on our risk assessment to other species, including livestock and ourselves. So that brings us to some of the things we can do, which is surveillance and management. And how are we doing so far? We good? Good? Parker's my gauge back there, so lucky you. Okay, so surveillance. This is kind of the first step, the preliminary step. And this is the ongoing and systematic and continuous collection, analysis, and interpretation of health data. And notice I said health data, so not necessarily specifically for one disease, because it has three different elements. So it can include environmental, it can include the susceptible host species, and it can include that disease agent. So it's kind of three-pronged, focusing at, what does that sound like? Yeah, I heard somebody, the epidemiology or the triad, the triangle, right, that causes disease. So it's three-pronged, and it can take a couple of different forms. So we have live versus post-mortem surveillance. Live would obviously be on live animals. Post-mortem is on dead animals. So we don't necessarily have to wait till we see a mass die-off and we're just collecting carcasses. The other kind of difference in surveillance comes from reactive versus proactive. So reactive would be we've seen an animal die, or we have a dead animal here, or we have a mass die-off. Let's go collect those, cut it open, see what happened. So we're reacting to disease. Proactive would be going out and sampling animals, regardless of whether we see disease in them or not. So we're looking at how many animals have it. Maybe it's there and we don't know it because it's in the subclinical phase. So we're going out and looking for it. We're not just waiting for it to happen. And all of this is important because, as you might have guessed, knowledge, knowing about these diseases, knowing where they're happening, is our first line of defense. So this is arguably the most important step, knowing what's going on. So once we have those samples, or we have that animal, we have that carcass, whatever, we have to figure out what's wrong. And there's a couple, or well, 5 different diagnostic categories that can help us do this. I realize this is a lot. I just wanted to give you some examples of the type of tests this might include, but really just focus on the main categories. So we have chemistry. That comes up a lot for toxicology, for body condition, nutrition, things like that. Microbiology, you're culturing things, you're doing genetic analysis to see what it is. Parasitology and virology, y'all can guess what those relate to, right? It's in the name. And then probably the most important one and the most easily accessible one is going to be necropsy and pathology. So necropsy, that's an autopsy for an animal. So we have a carcass, we're going to cut it open, we're going to visually assess what's going on there. We may take x-rays, we may take photos, and then we will take tissue samples. And this will go off for histology or pathology, where they're going to look at the cellular level, what's going on in those tissues. Any questions on those? Okay. Good deal. So now we have figured out what's wrong with our animal. Now we can move on to management. So management, remembering our terminology, this is at the population level, not the individual level, but it can include the individual level, which is treatment. So at the population level, we can do several options for management. We can target the specific disease agent. So we can target the influenza, or we can target the fungus that causes SFD. We can manage the host population. So for CWD, that might be reducing population densities of that host population. We can perform habitat management activities. So we can, so maybe say West Nile, which is found in mosquitoes. We can maybe do things to the habitat to promote or to reduce the amount of mosquitoes on the landscape. We can get rid of pools of water, things like that, so that there's less mosquitoes and thus less transmission. And we can also manage our own activities or our own involvement, which is particularly important for diseases that affect us, that you mentioned. So what can we do? I kind of already touched on this. Treatment at the individual level is potentially an option in some cases, but on a large scale, it is generally not possible. You can't administer enough antifungal agents across one county of Kentucky, let alone the entire state, let alone the entire US, right? So treatment of an individual is very, very spotty and isolated. We can do things on a larger scale like immunization. So those oral rabies programs, vaccine programs that I mentioned with those kind of barriers, we can do that. We can reduce populations. So thinking for CWD, we can modify the habitat. And then lastly, we can help restrict the movement of animals, either through those barriers that we talked about. So maybe roads can be barriers. We can also restrict our movement of animals. So we can impose regulations that prevent us from moving deer from one state to another, things like that. And you'll notice all of these, I mean, it's not a very big list, but out of the options that I did list, there was nowhere on that list was eradication. That might be possible in isolated cases, but again, at the large scale, it's not. So generally, we strive for control, which sounds a lot like that firefighter perspective. However, despite what's actually possible, our gold standard and what we always strive for is going to be prevention. That's the key. Because if it's not here, if we prevent it from coming, we don't have to have a response, right? And of course, management is not possible without public involvement because, you remember? Ownership.
Ownership, yeah.
Yeah, there you go. I jumped you on that one. It's okay. Because wildlife is a public resource. So we need all of the owners of wildlife to be on board. And this is mainly in the form of education and transparency. So if they know about what we're doing and they know about the concern, then maybe the public's more willing to help us out and be compliant with some of our management strategies. So this might look like increased reporting because they know about our surveillance programs, or we may gain access to samples or diseased individuals. We can increase the effectiveness of our regulations and management because they're more willing to participate. And through all of this, we can help reduce the risk to humans because now maybe they know that they should be concerned about this. But just like with everything, there's a but. You have to be careful about how we present this information. We don't want to scare people unnecessarily. We don't want to talk too technically that they are going to not pay attention or not understand what we're trying to do. So you have to be strategic, right? And there's a couple of different levels that this occurs at. So at an international level, it might be things like the UN or the OIE, which is the World Organization for Animal Health. So they are monitoring and collecting surveillance data worldwide on certain diseases and helping to kind of promote international level regulations. At the national level, we have multiple agencies or labs that are responsible for surveillance, for diagnostics, and for helping out with management. In Kentucky, one of the most important ones for us is going to be the Southeastern Cooperative Wildlife Disease Study, or SCWDS, which is in Georgia, because that's the closest to us. So any kind of surveillance and diagnostic measures generally get sent there. However, one of the big national ones is this USGS, National Wildlife Health Center up in Wisconsin. They do a lot of research, they do a lot of surveillance. And then of course, USDA APHIS and their National Resource or Research Center in Colorado. Moving one step smaller, most states like Kentucky have a state wildlife agency that also has disease surveillance and management, either kind of their own separate section or included in their routine wildlife management. So Kentucky has this, they have a state wildlife veterinarian, and she works closely not only with all of the wildlife managers and biologists around Kentucky, but also with public health departments, the Department of Agriculture, regular veterinarians. So it's kind of a big network, but all happening at multiple scales. Okay. Any questions? Is it all making sense? Nothing's like gibberish to people? Good, okay. Okay, so let's move into our last section. The wildlife disease is all well and good for the wildlife, but why should we care about it? I mean, we do because we like animals and wildlife, but why should the world in general care about it? And they should, because there can be major implications for humans. And before we jump into the specifics, just have a couple of general terms that hopefully you guys are all familiar with. Zoonosis, which is when an animal spreads disease to a human. So things like rabies would be an example of this. An epidemic is when a disease affects many individuals within an area or population at one time. So kind of isolated to a geographic region, but occurring at a greatly or a significantly greater occurrence than we think it should or than we're used to. And if we're unlucky enough, it gets bigger and it expands beyond that geographic area or population and it becomes a pandemic. How many of you have heard of that one? Every hand in this room should be up, right? That's the reason for this. So The biggest risk wildlife disease poses to humans falls into this category of emerging infectious diseases, or EIDs. These are known pathogens that are increasing in incidence or geographic range, or new pathogens, things we have never ever seen before and have no idea what to do about. These are important, especially considering wildlife disease, because 75%, 3/4 of them are zoonotic, meaning they can transfer from humans to animals and vice versa. And most of them, up to almost 2/3, are found in wildlife or originate in wildlife. That's a pretty alarming number and a pretty big potential for disease. However, the risk can be different at different scales, which is why we have all of those agencies at different levels, right? So we're covering the global scale at international levels, regional might be those national labs, and then local might be your Kentucky wildlife veterinarian. However, at each of these levels, there are two types of diseases or disease transmission methods that have a higher risk than any others. And those are viruses, particularly mammalian viruses, because they originate in mammals and we are also mammals. So it's a little easier to make that jump when you have a lot of the same biology. Excuse me. And then the second one that's at a higher risk, no matter the scale, is vector-borne diseases. And so these would be things like tick-borne diseases or rabies, something that's passed by another organism. And that's mainly due to things like climate change and kind of global development or travel, right? So if it's warmer longer, ticks can stay out longer, thus there's a greater potential for transmission. And of course, I can't get out of this lecture without bringing up the big one, which is COVID-19. We're currently in this pandemic, but its source, its initial source is still unknown. There's ideas, but they haven't been confirmed yet. But despite this, and why it's called pandemic or viral pandemic is because it has had a huge geographical range in its impacts for humans. So this was last week, but this is cases in humans. It's from Johns Hopkins website. And we all know about this. You know, I think we just recently hit 5 million deaths worldwide, which is a very alarming number. But the reason I'm bringing it up is because it also can be catastrophic or found in non-human species. So it's zoonotic, right? It's one of those things that impacts us. It's an EID because it was novel. We didn't know about it. Excuse me, and it expanded rapidly geographically. And you'll notice here, so kind of ignore the numbers. This is a paper that went up for publication back in the summer. So that's what the numbers are based on. But what I really wanted you to notice here is how many different species it's also found in. So we have domestic species, right? We have cats and dogs and ferrets. But we also have captive wildlife. So we have big cats, we have gorillas, right? Everyone probably heard about the lions and tigers at the Bronx Zoo that tested positive. Many zoos are also starting to look at potential vaccination programs for species at risk or endangered species. But one of the things I also wanted to point out is that it is showing up in wild populations too, which, scary, because it becomes infinitely harder to control, right, for the reasons we already talked about. So it showed up in one wild mink. That was due to contact with farmed mink that have been very devastated by this disease. This one, I believe, occurred in Utah. White-tailed deer, there are wild white-tailed deer. There's a big population of them in Michigan that have tested positive, confirmed tested positive for COVID. We have no idea how they got it. And then I threw this one up here, which was black bears. Because I was listening to a presentation at the end of the summer, and the speaker said, "Oh, and we found some positive tests in black bears." I'm thinking, what the heck? That's weird. You know, they're not like primates. They're not very similar to us, but they are mammals. And they went back and they retested them. And it turns out that it wasn't actually COVID-19, but it was another coronavirus. So I put this up here to illustrate that not only is this coronavirus, the SARS-CoV-2, found in wild and domestic species, but there's a lot of other coronaviruses that are normal in wild populations that may look or have similar symptoms or transmission to COVID. So we have all these EIDs, we have COVID, we have a global pandemic. How the heck do we approach that, right? We can't just do it from a wildlife perspective or a human medicine perspective, but rather we have to approach it using this new-ish management tool called One Health here. It's not new per se, so it started out in more focused concepts like one medicine or one world, one health. So kind of looking at the global human medicine environment. But really, One Health came about in 2004 when talking about the SARS epidemic in the early 2000s. Have anybody heard about that one? A couple people, okay. So that is very similar to COVID. It's actually the precursor, really, kind of a precursor to COVID. It even has very similar name. So SARS is severe acute respiratory syndrome and functions a lot like COVID, although the transmission mode and the onset of transmissible disease and symptoms is a little bit different. So that's when we started to see this management tool pop up. And it's only kind of gained in traction and awareness over the years, I guess, because it has two main characteristics. One is that it recognizes this, this kind of connection between animal health, human health, and environmental health. So we're kind of bringing in aspects of that epidemiological triad, right? And we're looking at more than just one narrow focus of this one disease in this animal. It also acknowledges animals as early warning signs. So for those EIDs that have 75% are zoonotic, with most of them originating wildlife, we're looking at wildlife first to see where those pop up, to see where humans might be at risk. One last benefit, and why you'll see a lot of programs are moving to One Health. It's very trendy now, but it's very important, is because it promotes communication and collaboration, right? Which leads to inevitable, I guess, success or increased success of public health interventions. And this is mainly due to it bringing in a lot more than just, like we talked about, more than wildlife managers. So you have the standard human health would be doctors, nurses, public health officials, animal health, you have veterinarians and wildlife, and then of course environmental scientists. But it also brings in kind of this new group. So police officers, EMS people, policymakers. So how can we regulate to prevent things? People, public members of the community as well, pet owners. I mean, the list goes on and on. I just named a few. So it's really a bigger, more coordinated response across many different fields. And that is because the main goal of it is to target disease threats before they become a global problem, before they cost us billions and billions, trillions of dollars, and before they cost us lots and lots of lives. So that is all that I have for you guys. I know I kind of rushed through a lot of that information. Does anyone have any final questions before we go? Okay, awesome. Well, thank you guys. The lecture is on Canvas, I guess. Yeah, have a good one. You're welcome.
Before switch majors, this is what I was doing, but humanities. So I've seen all this before. Microbiology and epidemiology.
You're welcome. Have a good one, guys.
Thank you.
Yeah, thank you guys. Have a good one.
Good.